It is I once again, Mr. Brothwell. It's still Friday, but you're going to be watching this on Tuesday, and this is dealing with immune responses. So, what we're going to try again to answer is the role of cell signaling in immune responses. And again, hello, Mr. Ng, Tien, sit down. Pause this as is necessary. So, when we look at cell signaling, the entire process turns out to be the exact same every single time. We're going to have a signal that's going to bind to a receptor. When it binds to the receptor, that's going to set off a transduction cascade, whether that's inside the cell or not, or it's going to be inside the cell, but whether that's affecting the nucleus or not depends on the nature of the receptor and the signal molecule. And eventually what that's going to do is trigger some type of response. Either we're going to have the cell change its behavior or it's going to change its chemistry, which is to say there's a genetic change occurring. So again, immunity and defense turn out to be two very different things. Defense is very non-specific, and it's just kind of, hey, just let's stop whatever it could be from harming the cell. Whereas immunity is actually something that requires a very specific response. So in order for us to talk about immunity, we need to deal with some terms. Those two terms would be antigens and pathogens. Anything that binds to a receptor that triggers some type of immune response we call an antigen. Some, or a good chunk of antigens turn out to not seek to cause you harm. There are, however, antigens that will cause you harm. Those antigens, the ones that will cause you harm, we give the name pathogen to. So antigens just trigger an immune response. Pathogens are antigens that also will cause disease or cause you harm. Not all pathogens are antigens, but not all antigens are pathogens. This unfortunately requires an unbelievably high degree of specificity, meaning we need to be able to recognize what these things are that are binding to us, and that is an insanely tall order. So it turns out the cell has a mechanism to do that. If you recall, when we go through and express genes, we being eukaryotic organisms, going through that process of expressing genes, what we have to do is we go from DNA to RNA and RNA eventually into protein. But for us, we need to modify our RNA. We have a pre-RNA and we have a modified RNA. In that pre-RNA, we have introns and exons. What we need to do is splice out the introns and leave only exons. Sorry, needing to breathe for a moment. So when you look at one of these genes, especially an immune gene, one that's going to make receptors or other proteins that we'll call immunoglobulins, what we need to be able to do is create a whole bunch of different types of proteins, because proteins are going to be what does the recognition. So in doing this, what we have are the production of the proteins that need to have variation built into them. And it turns out we have well over 1,000 varieties of exons that we can pick from. And as long as we end up selecting the correct numbers of exons, not the correct types, but the correct numbers, we can build a functioning protein. The result is, if I have 1,000 different options I can select from and I need 5 of them ultimately, that's 5 to the 1,000th power in terms of different things I could have here. What does that result in? It's millions, maybe billions of possible combinations of these various exons in order for us to build some type of functioning protein. If this is confusing to you, we're going to do it again, so don't worry. Point is, the way that we build these proteins allows for many, many, many more opportunities in terms of structures than we're probably ever going to deal with. Also, when we deal with immunity, especially with animals, we have to look at cells. And the cells that are responsible for immunity inside of animals are called lymphocytes. They are a type of white blood cell. They are very easy to identify. They have an insanely large nucleus comparative to the outside, comparatively speaking to their cytoplasm. They're divided up into two categories, B cells and T cells. You can't tell them apart by looking at them. You can only tell that they're different based upon their functionality. All that said, we can now deal with the types of immune responses. There are two. One of them is called a cell-mediated response. The other one is called a humoral response. The cell-mediated response only utilizes the cells that we call T cells. So the entire time, I don't think I have one. Okay, I do. So what we happen to have is we have some type of cell that has an antigen on it. So it's somehow presenting an antigen. That cell is going to be recognized by a T cell. So I'm going to just kind of flip back and forth. So I have some cell, it's going to be recognized by a T cell. The T cell is going to be able to recognize that specific antigen. Not all T cells will recognize it. How do they know? Because of all this variation that we build in. So the T cells have the ability to identify a very specific type of antigen, and only that type of antigen. When they do recognize each other, meaning the T cell recognizes an antigen cell, what it's going to do is this T cell becomes activated. It's going to actually undergo cellular or cell signaling, because we have the binding of a signal. It's going to cause signal transduction. It's going to cause different behavior. That behavior is what we call a cytotoxic T cell. And what those will do is they're going to go around and hunt down any cell that has the protein, has the antigen. And what they'll do is they're going to release chemicals that cause the cell to explode. Oh, well, that's fun. Yeah, yay. Because it's always nice to know that, yay, my mind is going the right way. So when these activated T cells find a cell that happens to have those antigens, it will attack in such a way that it causes that cell with the antigens to lyse. It'll explode on us. What cells do these target? These target virus cells and cancer cells. One thing that was shown in this picture is there's going to be memory made. That's going to be important later on. When we have this interaction, when finally a T cell gets to recognize an antigen, the result is memory. There's going to be something to remember. We're making a cell that's not going to go out and attack. We have a cell that's job is to stick around to say, I exist because this antigen was there before. Those cells are called helper T cells. And those helper T cells turn out to cause all sorts of things. So in part, they become those killer cells, but they also are used in the other half of immunity that we call humoral immunity. The way that the helper T cells do their job is they actually need to find an antigen. The cells that present the antigens are called antigen-presenting cells, APCs. Those could be a cell that's infected, or it could be a cell, like a phagocyte, that goes and eats other cells and says, hey, I ate this weird thing. What do you think it is? Either case turns out to work for being an antigen-presenting cell. The helper T cell recognizes, or a helper T cell will happen to recognize, hopefully, the antigen, and the result will be we'll get a response. Those helper T cells will also go around and they were going to sit there and try and find the other type of lymphocyte, which was the B lymphocyte, because the B lymphocyte is the one that's responsible for the humoral response. It's referred to as humoral, namely because it produces a chemical. And chemicals don't look like cells, they look like liquid. And liquids are called humors. So what we're going to have is we're going to have our antigen-presenting cell tell the a specific helper T cell, that specific helper T cell will find a specific B cell that they will match with. And what that binding will do is it's going to trigger cell signaling inside of the B cell. That B cell is going to differentiate. It's actually going to make a cellular response. And that cellular response is they become what we call plasma cells. These plasma cells differ from normal B cells in that they have a lot of ER in them, a lot of endoplasmic reticulum. Why would that be the case? Because endoplasmic reticulum is what you need in order to produce protein. The binding of the activated helper T cell to the B cell is also going to trigger protein production. It's going to trigger the central dogma of molecular biology. We're going to have transcription and we're going to have translation. What they're going to be producing are little chemicals. Those chemicals are called immunoglobulins. But if we happen to know what that immunoglobulin is going against, we know the antigen to the immunoglobulin, we give it the word antibody. All antibodies are immunoglobulins. Not all immunoglobulins are antibodies. We classify it as an antibody if we know, if someone knows what that immunoglobulin is going to react with, why it is there. When we look at those antibodies, they turn out to have various differing shapes. There are 5 classes of immunoglobulins, as you can clearly see here. Each one does something different, but they have similar parts, meaning they're built the same way, but the ends of them are always going to be different. The ends of these things are always different, and that is where the uniqueness of this system turns out to be. The shape is the same, but the ends recognize different antigens. So you have antigen presenting cell, it warns a helper T cell. Helper T cell goes, warns a B cell goes, activates, turns into a plasma cell, starts secreting these antibodies, these drones. What these drones can do that the cytotoxic T cells, the cellular mediated response, can't do, is they can have one cell that can cause lots of damage. The cell mediated response is cell-on-cell combat. The humoral response is you have one cell secreting lots of little drones, and those drones are going to cause all sorts of trouble. So among the things that those antibody antigen combinations, those, I don't know why I'm drawing blanks on words, when they combine, when they react, when they bind, what you can create are all sorts of things. So just going around the picture, you could actually prevent a virus from being able to get into a cell, or you can stop receptors, or you can make it so the cell isn't able to function, or it can trigger a defense response called complement. It's actually one of the pictures I showed you in the last slide, but I didn't talk to you about it. Complement is a whole bunch of little transformers inside of your bloodstream that form holes, and then they explode cells, because that's what we like to do. What you could also do is it could help trigger cell-mediated responses. It's kind of like saying, hey, eat over here, or it could cause phagocytosis. Actually, it's called opsonization, which is saying, hey, eat this thing, or just their mere presence will trigger phagocytosis. Or what you could do is cause things to precipitate out, or will trigger an inflammatory response. There's all sorts of things that these antibodies do when they bind to their antigens. Cell-mediated response, cell-on-cell combat. Humoral response, drone strikes from everywhere. So needless to say, this is kind of a very big, powerful tool. And again, all of this works because a very specific antigen was recognized by a very specific T cell, which told a very specific B cell, which is producing a very specific immunoglobulin that we call an antibody. Without that specificity, which came from this horrible slide that freaked you all out, which is when we make our protein, when we finish making our mRNA, we're going to take from all the thousands of different exons that exist, we're only going to use a handful of them. What I can get is specificity, meaning if I'm making an antibody that goes against Tien with whatever Tien is wearing today, but if Tien were to comb his hair, automatically the antibody does not work anymore because Tien has changed. If Tien changed his clothes or changed like his watch, or he got a cut on his skin, he has changed, specificity has changed. The antibody that worked once does not work anymore because he changed. That's how specific these things turn out to be. The B cells and the T cells all turn out to make memory, and the reason why is for repeat exposure. If you get sick once, okay, we have to go through and we have to figure out that, oh, okay, here's what we need to do now. But if it happens to you again, you want to be ready. Clearly this thing got inside of you 2 times in a row. This might be a bad thing. So we need to be ready to take it out. So the result is the first exposure, you make so many antibodies, but then we produce memory. The second exposure, you are ready for the attack. We don't need to go through that antigen presenting cell, T cell, B cell stuff. We can go straight from a memory B cell to producing antibodies. We can skip a few steps. And the result is we actually produce a lot more antibodies. We get a lot more of a response. The catch with this is it's going to be a more severe response because more antibodies equals more inflammation, more fever, more phagocytosis, more everything. So this is going to be a much more severe response, hopefully to take out whatever the antigen is. We learned of this and then we took advantage of it, and we call it variolation, or sometimes called vaccination. What we do in this is we're intentionally giving you an antigen so that your body produces memory. By giving you memory, what we're hoping is if you really do get this antigen in real life, this pathogen in real life, your body will not start with first exposure but start with second exposure. By doing that, you get a much bigger response, and hopefully you don't get sick and die from whatever this pathogen turns out to be. This only works, however, with DNA viruses or DNA-based organisms, things that don't mutate too much. RNA-based organisms, for reasons we'll talk about later, such as the cold or the flu, those fluctuate a lot, which means we actually have to have lots of vaccinations, or we need to be changing the vaccination all the time, which is why you get a flu shot every year. You don't just get one when you're a baby and you're done. Each year the flu is changing on us. So the result is last year's flu is not this year's flu. The cold you had 2 weeks ago is not the same cold that you're going to get next week. And the reason for that is they're made out of RNA for the genetic material, not DNA. Plants do a very similar version, or a very similar version to all of that. Theirs is referred to as SAR, systemic acquired resistance. And what that requires is we have the recognition of some type of protein. It's going to trigger a hypersensitive response, which works for anything, including wounding. But what those signals will also do is start producing very specific chemicals meant to shut down whatever triggered this response in the first place. So the acquired resistance is very specific protein production meant to stop that particular, that particular antigen that triggered the cell signaling cascade and that also triggered SAR. We don't know as much about this as we do animal immune systems, and that's just because we're selfish. But plants have something insanely similar, and I'm sure the more people study this, the more we're going to come to understand it, much like how we're understanding plant intelligence a lot more.